Good morning, everyone. It is my pleasure to be part of the breast cancer webinar in its 10th edition. My name is Kholoud Arafa. I have attained my PhD recently in the field of nanomedicine. I am currently affiliated to Zewail City for Science and Technology in Egypt. Today, I'm gonna introduce you to a very interesting topic, tackling the sequential and mitochondrial targeting of breast cancer through using partial nano drug delivery system. In my presentation, I will give a brief introduction about mitochondrial medicine, and then I will introduce you to the scope of work, the methodological approach that we have deployed, going onward through the results and discussion section, and finally the conclusion. So, what is cancer? Cancer is an enemy, and it is a group of disease that affects both genders equally, affecting so many different organs. Cancer incidence globally has been on the rise, according to the WHO reports and the Global Cancer Observatory records for year 2020. Breast cancer constitutes almost 12% of all the new cases reported, and it caused about 7% of the entire deaths for this year. In Egypt, breast cancer comes in the second place of all the newly reported cases of cancer, according to the GloboCan for year 2018. Given the daunting number of cancer occurrences globally, it has been very important to understand the nature of cancer as a disease. And cancer has so many different properties than normal cells, including its ability to evade growth suppressor signals, its ability to resist cell death and apoptosis, its ability to proliferate in an uncontrollable manner, deviating from or evading cell cycle checkpoints. It also has the ability to support the formation of new blood vessels and to induce what is so-called angiogenesis, and to form distant replication sites through invasion and metastasis. All these properties make it a very intricate disease and a complex disease, and all these properties were points of care and research for the treatment of cancer. However, there is a paradigm shift in cancer research now, focusing more on the metabolic phenotype of cancer. It started back in 1924 when Dr. Warburg reported the increased glycolytic activity of cancer cells relative to normal cells. And through the years, it has been shown that so many mutations affecting the metabolic enzymes, including SDH and FH, were known to be implicated in carcinogenesis. Also, specifically in stubborn cancer types like glioblastoma, IDH or isocytrate dehydrogenase, a metabolic enzyme, again has been reported to play a very important role in the pathogenesis, all of which collectively pinpoints dysfunctions of mitochondria to be implicated in carcinogenesis. This led to the focus of Dr. Weinberg and his group again, and putting forward a new, fresh point of view of the hallmarks of cancer, including a dysregulated metabolism and cellular energetics as one of the very important emerging hallmarks of cancer. So if we look at the mitochondrial structure, as we all know, it is made of an outer membrane, an inner membrane, and the matrices where all the metabolic functions occur. Looking just at the structure of the mitochondria, for the normal mitochondria, we can clearly see the crystal that forms within the mitochondrial matrix, as compared to the mitochondria attained from cancerous cells, specifically GBM or glioblastoma cells. We can see that there is a significant amount of crystallysis and absence of the matrices, and this tells you that there is a structural dysfunction in the mitochondria that makes it very distinctive than its normal counterparts. Seeing this striking difference between the structure of mitochondria in the cancer cell versus its normal counterparts, it was very pressing to understand whether this dysfunction is a consequence or a cause of tumorigenesis. And a very nice approach to answer this question was to have two different cells, one containing normal mitochondria and cancer originated cells. On the other hand, we have a cell that contains tumorigenic mitochondria while having normal cells, and to see and monitor the phenotype of the progeny. And it was concluded that having a tumorigenic mitochondria was the cause of propagating a tumor cell phenotype, which implicates the mitochondria more as a causative rather than a consequence of tumor formation. And this seems to be logical, as mitochondria plays vast processes and is part of vast processes in the cellular homeostasis, including ATP production, ROS signaling, metabolic flexibility, being involved in response to therapy. Again, all of these parts would eventually help into the transformation of a normal cell into cancerous cell, and making it harder to vanquish or to treat such cancer cells. Compiled and collectively, this has directed the efforts of so many scientists through years to focus more on mitochondria as a druggable drug target. It can be seen in the figure that there are so many subcellular organs or organelles that can be targeted for therapeutic purposes, including nucleus, endoplasmic reticulum, and Golgi apparatus. And it can be seen that mitochondria is now, or mitochondrial medicine now, is on the rise. With the concept of mitochondrial targeting occurring, it is very important that we produce or develop spatially selective targeted chemotherapeutic agents. One of the drawbacks for conventional chemotherapy is that it affects both cancerous population as well as a healthy population equally, and this is on the cell level, and when it comes to the organelle level, it's the same, that the random distribution of the chemotherapeutic agent within the organelles within the cell is again a very deleterious as a concept. So with the rise of nanomedicine field and the application of nanotechnology in the biomedical field, several platforms and drug delivery systems came into display, including passive targeting of nanoparticles starting in 1986, and the functionalization of the nanoparticles to produce active targeted nanoparticles in 2002 led eventually to the preparation and fabrication of mitochondrial targeted liposomes in 2003, and this opened so many venues for nanoscale drug delivery system directed towards the mitochondria, and it was the real start of growing this field from 2012 on. To achieve mitochondrial targeting, several ligands have been used, starting with 2000s. DQazomes has been deployed for targeting the mitochondria. Onward to 2003, TPP, which is a different class of ligands, has been deployed to develop liposomal-based drug delivery systems. And in 2012, biodegradable polymeric nanoparticles has been developed to be targeted to the mitochondria. Knowing that there are several targeting ligands, what are the biotargets of the mitochondria? There are so many biotargets that could be hit by the chemotherapeutic agents, including those affecting the metabolic pathways, also the ETC chain that is responsible for ATP production and oxidative phosphorylation to produce ATP, the mitochondrial DNA, which is an organelle-specific piece of DNA, those enzymes that are implicated in redox pathways and redox homeostasis, and those molecules that are important for the induction of apoptosis, especially mitochondria-based apoptosis or intrinsic pathways. So, by nature, there are a group of drugs that could be titled or named mitoactive chemotherapeutic agents. In our work, we have chosen doxorubicin as the mitoactive chemotherapeutic agent. Doxorubicin can affect the mitochondria in different aspects, including manipulating the calcium homeostasis, propagating lipid peroxidation, the induction of RAS production through Fenton reactions, the inhibition of DNA repair both at the nuclear level and mitochondrial level by affecting topoisomerase isoenzymes 2 and 1, and inducing mitochondrial DNA damage. All of these eventually will lead to bioenergetic imbalance and cellular death. There are a wide array of molecules that could be used as mitotropics and ligands for mitochondrial targeting, amongst which was TPP, and it is the ligand of a choice for our work. Why did we choose TPP? TPP is short for triphenylphosphoname cation. As we can see from the structure, it contains 3 phenyl groups and a phosphoname containing group. Due to the hydrophobicity of TPP, it can easily pass the cell membrane, and relying on the difference between the mitochondrial membrane potential and the plasma membrane potential, TPP can accumulate more inside the mitochondria, and there is a difference between the mitochondrial membrane potential and within cancer cells and normal cells, making it of therapeutic selectivity, because the mitochondrial membrane potential in cancer cells is known to be more negative, leading to 100 to 500 fold increase of accumulation of TPP in cancer cells. So all of these leads to a therapeutic selectivity and towards the mitochondria. A recent approach that has been adopted for targeting of nanoparticles is called hierarchical targeting of nanoparticles, which relies on first introducing the nanoparticles specifically to the cancerous niche through cellular targeting, followed by organelle targeting, and in our case it would be the mitochondria, with the hope to induce apoptotic cell death. So the scope of work is to achieve good physicochemical stability of the nanoscale drug delivery system, and to enhance the encapsulation of the anti-cancer drug. In our case, it's doxorubicin, and to or relying on sequential targeting to develop um, uh, um, a drug delivery system that would eventually attain improved pharmacokinetic and pharmacodynamic properties. There are a platform of a plethora of smart design nanocarriers. In our work, we will rely on polymeric nanoparticles. Our targeting ligand will be the small molecule TPP, as well as a polymer, hyaluronic acid, and we will rely on enzymatic and pH stimuli to be our stimuli or trigger of dismantling the nanoparticle, and our aim would be killing cancerous cells selectively. So, as a general approach, we have first synthesized our double-stage targeted drug delivery system with a shell that is made of, with a core that is made of chitosan-based nanoparticles loaded with doxorubicin, and this forms the core, and eventually being coated by hyaluronic acid. Then after the physicochemical characterization of the developed nanoparticles, we have applied these nanoparticles on breast cancer cell lines to validate its biological activity. Once we found promising results on the in vitro level, we've gone through the in vivo level to make sure that it is functional in physiological mimicking conditions. So first we prepared our drug, which is a conjugate of doxorubicin mitotropic form, in which we have linked chemically or covalently TPP cation to doxorubicin through EDC reaction. We have validated the formation of the conjugate through FTIR and NMR approaches, and it was evident that there is the triphenyl system being part of the doxorubicin as shown in the NMR chart. After validating drug conjugate formation, now we have started the nanoparticle preparation. We have relied on ionic gelation for the preparation of the chitosan-based core, followed by polycomplexation for the formation of the core shell nanoparticle. For the ionic gelation, we have mixed or stirred the drug conjugate with tripolyphosphate as a cross-linking agent. Then adding this solution to chitosan solution, dropwise addition, with the aim of forming drug-loaded chitosan nanoparticles cross-linked with the tripolyphosphate. After the formation of chitosan nanoparticle as a core, we have added hyaluronic acid dropwise to the chitosan solution. Now the chitosan nanoparticulate nanosuspension under stirring, relying on the polycomplexation that occurs between the negatively charged hyaluronic acid and the positively charged chitosan. We aimed at the formation of a core shell nanoparticle. We have optimized so many parameters, including the drug concentration, the polymers ratio to each other, and varying the drug type as well, and measuring how it affects several factors, including particle size, polydispersity index, and zeta potential. Based on the outcomes of different trials, accounting for 16 different formula, we have obtained optimum nanoparticles denoted F14 and F15, which contains 20% of the drug to cytosine weight per weight, and it has a hyaluronic to cytosine ratio varying between 0.5 to 1 and 1 to 1, with the particle size in the 300s to 400 nanometer range, and a positive zeta potential, which is actually favorable for cancer cell targeting, and attaining actually the highest loading capacity, which was very important parameter for us to ensure proper loading of the drug within the formed nanoparticles. The successful formation of nanoparticle could be depicted from the TEM images, and it can be seen that it is of a spherical shape and a monodispersed monopopulation with the size of 200 amphores, and also the FTIR confirms the successful formation. Having the nanoparticles successfully made, we wanted to ensure the proper release of the drug towards the cancerous niche rather being dissipated in the blood. So we have attempted the release in different pHs, and as it can be seen from the figure that higher release rates were evident in acidic pH rather than in physiological pHs, which again confirms the selectivity of drug release. We have made release kinetics, and we found that most of our formulation followed the Baker-Lonsdale mathematical equations. To validate the biological effectiveness of our nanoparticles, we could see we have implemented cytotoxicity experiments, and we could see that the IC50 of the mitochondrial nanoparticle is close to the free drug, but again with the higher affinity towards cancer cells, which is more favorable for selectivity of drug release and evading the undesired adverse effects. To elucidate the mechanism of cell death, we have implemented apoptotic analysis, and we could see that the optimum formula could induce apoptosis 4 folds higher or 8 folds higher than the untreated control, while the necrosis also could be seen to be significantly higher than untreated control. It is evident that our nano formula could induce G2M arrest, cell cycle arrest, and pre-G1 cellular redistribution, again confirming that apoptosis is the mechanism of cell death. Fluorescence imaging further confirmed the localization of our nanoparticles to the mitochondria, as can be seen by the yellow areas in the merged image. All of these collectively confirmed the biological effectiveness of our nanoparticles in killing breast cancer cell lines in vitro. So based on the promising results, we moved onward with the in vivo biochemical assays, and we have seen that the nanoparticulate treated animal groups attained lower tumor volume compared to the non-treated control groups, and the tumor weight was significantly decreased as well. As for the parameters of hepatic function, it could be seen that the nanoparticulate system could retrieve the hepatic integrity and functionality, um, uh, as uh, as compared to the free drug, as can be seen in the ALT protein concentrations. For the histological examination, it could be seen that treatment of the treatment of animals with DOX-loaded, DOX-TPP-loaded nanoparticle could retrieve the normal histological structure of the liver as compared to the free drug or uncontrolled animal group. Again, showing the high potential of the nanoparticle to reduce the toxicity of the drug and to treat cancer as well. To further confirm that apoptosis is implicated in the cytotoxicity induced by the drug conjugate, we have analyzed immunohistochemically the levels of caspase-3 protein, and as it is evident from the images that there is an upregulation in the caspase-3 protein in nanoparticulate treated group relative to control group. So conclusively, the optimized nano drug delivery system demonstrated high physicochemical stability and homogeneous morphology. TPP conjugate prodrugs as well as nano drug delivery system proved to accumulate in the mitochondria. In vitro and in vivo biological assessment showed that TPP-linked nano drug delivery system exhibited higher therapeutic efficiency. By this, I would like to thank you for your time and to thank my team for helping me achieve such work, and I hope that it introduced you to the novel approach of developing mitochondrial targeting drug delivery system, and I would leave the floor for questions. Thank you again for this chance.